today's speaker, I'm going to give a very brief overview of Agilent biopharma workflows and some new exciting things that we're doing. And then we're going to have a talk by Harsha Gunawarda at Janssen, who's going to be talking about analytical innovations to speed up antibody characterization.
Thank you, Mike, for giving me this invitation to come and present here. And today I'll be talking, and I'm very excited to talk about some of the analytical innovations we've been doing at Janssen to speed up antibody characterization. So today I'll be talking about two disruptive technologies. To give a brief outline, I will describe the need for both speed and comprehensive characterization of biopharmaceutical, biotherapeutics in biopharmaceutical R&D. So my talk will be two parts, where first I will describe a new technology, which is known as mobile affinity selection chromatography. And this technology has been used to determine the aggregation and the titer of a biotherapeutic directly from a bioreactor. And we've also implemented very rapid, ultra-fast titer determination by extending this method for a very high throughput format. So to give an introduction to folks who are not too familiar with antibodies and the drug discovery and development process, so as we all know, monoclonal antibodies have been for a while. There are over 140 monoclonal antibodies that are used for both therapeutics as well as diagnostics since they were first introduced in the early 1980s. So more recently, over the last 10 years or so, we've seen the move from monoclonal antibodies to this new modality known as a bispecific antibody, or they can also be multi-specific antibodies. So as the name implies, these modalities recognize more than one antigen simultaneously. In the case of the most popular CD3 redirectors, you're harnessing the power of the immune system to target cancer. So these antibodies don't come without problems. They are prone to aggregation, which is one of the major problems of these types of antibodies. They can also undergo chain mispairing because there's more than one heavy chain in this antibody, and clipping and insertions. On top of all these, we also see the common post-translational modifications, and also sometimes we can encounter unexpected or very unusual post-translational modifications. Then we also can introduce different small molecular drugs and conjugate them antibodies, and this creates this large number of proteoforms. So in order to tackle this proteoform diversity, we need to have faster and more comprehensive analytical methods. So just to give an outline, just to give the various stages of the drug discovery and development pipeline, you can see there are various discovery stages where antibodies are generated, and we end up with a few antibodies that enter cell line development, where we also produce a large number. We start off with a large number of clones, and then we enter a stage where we are sending a single clone into late phase development. So may it be generating large number of molecules or large number of clones, analytical cannot be the bottleneck. So how do we overcome these bottlenecks is we at Janssen in our group have relied on automation. I won't be going through most of it. I'll just give you a brief outline of this end-to-end pipeline, where we start with the LIMS system as a sample management system, and then we have high throughput robotics to introduce samples directly onto the mass spectrometer. So there is no human intervention. And in addition to this rapid fire MS acquisition, which I've shown here, we have a number of mass specs that are connected to this automation. Once the data is generated, it's automatically swept into a vendor agnostic software that resides in the cloud, and data is processed and aggregated in an in-house knowledge base. And then the cycle gets completed where that information is passed on to the LIMS system, and the data management starts all over and the cycle is completed. So this is just a workflow that I'm describing, but there are various elements to this workflow where analytical methods, especially some disruptive methods, can help the throughput. So in the first part, I'll describe a technique known as mobile affinity selection chromatography. So this is a technique where you're using the column as a protein reactor, and the company known as Novilytic has introduced this technology, and they call it a proteometer. And they call this technology the canary in the fermenter. So the reason for this is they believe that this technology can address problems that are happening and be an early warning system for the analytical chemist to take corrective actions. So how does this work? Is in a normal chromatography, in a column chromatography, we have two phases, a stationary phase and a mobile phase. This chromatography has the third phase, which is the mobile affinity phase. So the mobile affinity phase is composed of an affinity selector, a reagent that binds with the monoclonal antibody in a 1-to-1 ratio. It's very selective. It does not bind any other protein, and it also has a fluorescent tag. So once the binding occurs in a 1-to-1 ratio, they get separated on a Sievin matrix, just like a size exclusion chromatography, and the complex is then detected using fluorescence. We also see free floating fluorescence reagent, which can also be detected later on as it separates. So one advantage of this technology is there is no column regeneration, there is no carryover, and you can introduce samples and analyze them without sample preparation from direct crude culture supernatants. So here's a result of this chromatogram that we obtain using these columns and this technology. Is we get a single peak of the monoclonal antibody in the presence of over 1,500 host cell proteins and various types of metabolites. So there is no purification. Not only can we obtain the monomeric species, to your left-hand side you see the inset of the higher molecular weight proteoforms. So we can determine both the titer using a calibration of different concentrations of the monoclonal antibody, as well as the aggregation amounts that are found in these samples. And to your right, you see a negative dip in the baseline, and that is when the fluorophore elutes out of the column. So I'll get into the details of seeing that negative dip. That is important to note in the things that I'm going to describe later. So we've taken this technology and examined daily time points over the course of a bioreactor run. So AMBER 15 bioreactors have 24 bioreactors in 250 milliliters, and we collect the supernatants, do not do any purification, and introduce them into this HPLC. In this case, it was the Agilent 1290. This technology comes as a kit, which has different reagents, columns, as well as the buffers. So one advantage of the Agilent 1290 is it can do serial injections using sample pretreatment option to do sequential injections. So this is the result we get for the 16 days, and you can see we can determine the monomer, dimer, trimer, tetramers within a 10-minute runtime. And then off of a calibration, we can determine the titer, which is shown in B in the inset for all 16 days. We can also obtain the peak areas of the dimer, trimer, and the tetramer, and at the bottom of that, we can calculate or estimate the amount of aggregates that are formed as individual species or as the sum of all those species, which we call the sum aggregation. So how does this titer compare with the traditional ways of determining protein A titer? So traditionally, titer is determined after a purification step of a protein A column. So post-purification after protein A, the titer is shown in red, and the proteometer titer or the mobile affinity selection chromatography titer is shown in blue. So you may note for two clones that are run in two bioreactors, they are very reproducible, and they seemed more reproducible than the protein A titer that we get. So once we've established this, now how can we increase the throughput of these analyses? So what's shown on the left is the experiment where we've done is to not use a column, but introduce these reagents with the antibody using flow injection. So mixing occurs, and we can get the detection of this fluorophore bound to the antibody, but we lose the aggregate information. We can perform this high throughput assay in less than 1 minute, but you lose the aggregate information. To circumvent this, what's shown on the right is how we've used the column now, but we are performing sequential or serial injections of this antibody sample with the reagents that are in the column. So the first peak is the sample, and the subsequent injections that you see after a time lag is the standard that we inject at various concentrations. So we can obtain a standard curve directly and estimate the amount of sample or the titer of an actual sample. So to the right of the last peak, the positive peak, we get these negative dips, which I alluded to earlier, becomes an issue when we have to monitor antibodies or do serial injections beyond that point in time. So how do we overcome this problem? Is we relied on column switching. So here is a format where we're using 2 pumps and 2 mobile affinity selection chromatography columns to do sequential injections, but we do that in conjunction with column switching. So we do this column switching every 1.4 minutes, and by doing that, we can roughly analyze about 40 samples per hour, which is about 1,000 samples per day. So the advantage of doing high throughput titer determination using the column is you can determine the titer as well as you can do product quality analysis, where you can obtain the aggregate levels as well as determine the titer simultaneously. So I hope I was able to demonstrate the use of this mobile affinity selection chromatography technology to determine titer and aggregation levels. In our hands, we find that this is highly reproducible and more reproducible than the traditional protein A post-purification titers that we obtain using the standard method. And we also demonstrate a very fast titer determination method using column switching.